Hello, everyone, and thank you for tuning in to my presentation today. My name is Albert Burgess-Hull, and I'm a postdoctoral fellow in the Real-World Assessment and Prediction Unit at the National Institute on Drug Abuse. Today, I'm going to be talking about research conducted within our lab that is focused on the development of an opiate craving taxonomy for patients in treatment for opiate use disorder. Now, the idea for the study I'll be focusing on today was drawn from findings from two ongoing efforts within our lab to leverage data drawn from ambulatory assessment methods to capture information about drug use behaviors in real time. Now, using these methods, we have attempted to identify generalizable associations between a patient's mental states, environmental exposure, physiological changes, and lapse to drug use during addiction treatment. More recently, our group has focused on the development of personalized computational models to predict moments of risk for individual patients in substance abuse treatment. Our eventual goal here is to develop a robust and dynamically updating framework that learns to identify watershed moments for an individual patient so that a tailored intervention can be delivered to these patients in real time. However, during both of these pursuits, we ran into slight problems which have slowed down our progress. One of these problems is we found that the presence of heterogeneity within our samples has had a significant influence on our ability to identify generalizable associations between important predictors and lapse and the development of robust and accurate prediction models. An example of this is presented here, which is taken from a recent study we published using examining the use of GPS data to predict future stress and craving levels for patients with opiate use disorder. In this study, we found that subgroups of patients within our sample displayed significantly different levels of self-reported craving and stress during treatment. And the subgroups we identified for stress are shown here on the left. Furthermore, differences in these longitudinal patterns had an important impact on the accuracy of our individualized prediction models, which is shown in the figure on the right. Now, the identification of longitudinal subgroups of patients was not the primary goal of this study. However, the differences we observed and the potential impact these groups might have on the development of more accurate prediction models suggested that we should examine this heterogeneity further. Thus, we conducted the current study, which focused on testing whether patients with opiate use disorder exhibited similar or unique patterns of opiate craving over the course of treatment. If there was evidence for heterogeneous patterns of craving, and we're expecting that we would find differences, we were also interested in examining whether the subgroups differed on commonly assessed treatment outcomes. Now, the data we used for the study was collected from 237 opiate-dependent individuals enrolled in a natural history study during agonist maintenance treatment in our research treatment lab in Baltimore, Maryland. To capture data on these participants' behaviors and environment in real time, participants carried smartphones, which we provided to them for up to 16 weeks. These smartphones alerted participants at 3 random times throughout their day to fill out a short survey about the participants' immediate surroundings and their momentary craving and stress levels. Participants were also asked to fill out surveys if they used any type of drug, excluding alcohol or tobacco during the day. During these event contingent surveys, participants also rated their craving levels and reported on their immediate surroundings. Now, to test for the presence of distinct craving subgroups, we first needed to compute a measure of craving during treatment. As a result of the EMA data collection methods, participants provided at least 3 EMA reports of craving each day. Due to the length of the study, we decided to compute an average daily craving variable for each participant, which ranged from 1 to 5. We then used growth mixture modeling, which is a longitudinal clustering method, which identifies the presence or absence of unobserved groupings of individuals to test for the presence of unobserved craving trajectory subgroups within our sample. Now, for those interested in hearing more about this method, I'm happy to answer questions during the question and answer session. However, I've included a bit about model fitting and selection at the bottom of the slide. Now, after fitting the models and comparing the relative fit, of each of the models against one another, we found that a forward trajectory subgroup model fit the data the best. Now, the shape of the different patterns of craving during treatment and the sizes of the 4 groups are shown in the figure in the table on the left. First, what can be readily seen is that the patients in opiate agonist maintenance treatment reported significantly different patterns of craving over the course of treatment. Looking at the proportion of patients assigned to each group, which is shown at the table on the bottom, we can see that the majority of patients reported low and stable levels of craving over the course of treatment, which is shown in the green trajectory. However, we can also see that the next largest group of patients, the blue trajectory, reported initially high levels of craving that increased as they progressed through treatment. We can also see from the figure that another group of patients, shown in the black trajectory, reported initially high levels of craving that rapidly declined during the first weeks of treatment, and a final group, shown in red, reported an inverse U-shaped type pattern of craving and cost treatment. So we found evidence for the presence of craving subgroups during treatment. We were next interested in how these longitudinal differences in craving might relate to treatment outcomes. We first examined whether there were differences between the type of treatment a particular subgroup may have received and the number of days a patient remained in the treatment study. Overall, we found that the main differences were observed for the low craving and the high and increasing craving subgroups. Comparing each of these subgroups to patients in all other subgroups, we found that patients receiving buprenorphine treatment were more likely to be in the high and increasing craving subgroup, and patients in the low craving subgroup lasted the longest in the treatment study. Finally, we also examined whether there were differences in drug use during treatment for the 4 craving trajectory subgroups. And again, we found that the majority of differences were observed for patients in the low craving and the high and increasing craving subgroups. Comparing the drug use levels of these subgroups to the drug use of patients in the other subgroups, we found that patients in the high and increasing craving subgroup reported the highest levels of any drug use, heroin use, and any opiate use. Conversely, patients in the low craving subgroup reported the lowest levels of any drug use, heroin use, and any opiate use. Now, summarizing our results, we found evidence for qualitatively distinct opiate craving trajectory subgroups among patients in opiate agonist maintenance treatment. Furthermore, differences in these longitudinal patterns of craving were related to clinically relevant treatment outcomes, including drug use during treatment and treatment dropout. The majority of patients reported low and stable levels of craving during treatment, and these patients reported the least amount of drug use and were the least likely to drop out. Finally, some patients did, however, report high and increased levels of craving during treatment, and these patients reported the highest amount of drug use, and furthermore, patients who received buprenorphine were most likely to be in this subgroup. Now, these findings need to be interpreted in the context of some limitations. First, the data used to test for the presence of craving subgroups was collected from a non-random sample of patients seeking treatment at our clinic. In addition, clustering methods such as growth mixture modeling are known to be sensitive to sample characteristics and distributional assumptions. So as a result of these limitations, these findings need to be replicated to determine if they generalize to non-urban or non-Baltimore populations and whether they are robust to different samples and distributional assumptions. However, considering these limitations are a few things that we can take away. First, craving heterogeneity is important to consider during treatment. Differences in craving not only will influence the patient's experience during treatment, but identifying high and low groups of cravers can illuminate non-responders and optimal treatment responders, which is what we found. Finally, the identification of robust classification schemes can inform the development of personalized treatments. And although I didn't present the data here, we have found that by using only a subset of variables from the Addiction Severity Index, we are able to currently correctly classify more than 85% of patients in the high and increasing craving subgroup before they initiate treatment. So to end, I'd like to thank the individuals in my lab. And if anyone would like to contact me with any questions, here's my email address. Thank you.